My presentation here at ICRASAT is on a systems genetics strategy that is actually applicable to any plant breeding problem. And we have used this approach to study a really important fungal disease in Africa, as well as the rest of the world, of maize plants. So the disease is grey leaf spot disease, and it's a foliar disease of the maize plants, which is widespread across the world. In Africa, it's a particular problem. And so in my presentation, I present 3 strategies that we employ to really understand the molecular basis of defense in maize plants to this pathogen. So the first approach is a QTL mapping approach. The second is a global gene expression co-expression analysis. And the third approach is an expression QTL analysis. And in my presentation, I will illustrate how we bring together all 3 of those technologies and data to complete a meta-analysis to obtain a better view of the responses of the maize plants to this disease. And so this is essentially a systems genetics approach where we are actually combining phenotypic data together with global gene expression data and genetic mapping data. And this allows us to actually get to the molecular basis of a disease response. And in our particular study, we firstly were able to map QTLs for resistance or susceptibility to the disease in a particular mapping population of maize. And from this results, which we obtained in several years of study in 5 different field sites in Southern Africa where the disease is a hotspot, we were able to identify these genomic regions or QTLs for resistance. And that work was published in BMC Genetics in 2014. And what we did in my presentation is to present data where we actually took the QTLs that we found in one particular season, and we actually extracted RNA from 100 of those recombinant inbred lines from the segregating population and analyzed the expression profiles across all of those 100 lines. So it's a large amount of data that we had to analyze, and then conducted co-expression analysis. So we actually were able to identify modules, groups of genes, which are co-expressed across the whole population. And by correlating that expression across the population with the disease scores of the 100 plants, we were able to identify those genes which are actually upregulated in the susceptible response to the pathogen. And what we did next was then to apply expression QTL mapping to the same gene expression data. And by combining both the co-expression data with the expression QTL mapping, we were able to show that there's actually a genetic basis for the co-expression that we observed. And so the implications of this study is that we obtain a much better understanding of the defense responses of maize plants to this particular pathogen, and that can help in future molecular breeding approaches. Indeed, we actually were able to integrate one of the QTLs into different maize backgrounds and show increased resistance to the pathogen. But importantly, this approach is very exciting, and we can use next generation sequencing technologies instead of microarrays to do the gene expression profiling on any agricultural crop, including the mandate crops here at ICRASAT. And this will also accelerate, hopefully, our understanding of defense responses in important crops and help in molecular breeding for improved crop production.